Hi everyone, this is our second revision video for mid-semester 1 or for the mid-semester 1 test, and we have stopped here last time at the end of the first video. Concerning the concept of the tendons, there are multiple questions that you might encounter on the test. For example, you might have a scientific term like fibrous connective tissue cords made mainly of collagen that connect the muscles to the bone, and therefore you have to identify the answer as the tendon. Or you might be asked to define a tendon. So when you define a tendon, there are certain key words that you have to take into consideration. And these terms or keywords are fibrous, because it's mainly made up of collagen fibers, and collagen fibers are nothing but collagen molecules that are highly contractile and elastic. You have to mention that the tendon consists of, or these fibers belong to connective tissue. So what kind of tissue does the tendon contain mainly? The tendon contains fibrous connective tissue. The other key word is cord, because tendons are cylindrical in cross-section, most of them, in contradistinction with the ligaments that are more or less flattened. And that's why we say that the tendons look like ropes, because they have a cylindrical shape or circular cross-section. I might ask you about the importance of the fact that the tendons are made up of collagen. Of course, collagen is a contractile protein. It is also an elastic protein, so it provides elasticity to the mainly to the tendon and to all the structures that contain collagen. Some tendons are exceptionally broad and flat. The main function of the tendon, if you're ever asked about the function, sometimes you have a big question, mention the function of each of the following. The main function of the tendon is to help attach muscles to the bones. And the elasticity of collagen protects the tendons because it allows the muscle to pull on the tendon if the tendon is an insertion without being torn. So the presence of elastic collagen inside the tendon is protective to the tendon. Justify the statement because tendons attach muscles to bone. And when tendons represent the insertion, then when the muscle contracts, there will be a pull on the insertion tendon to pull the bone upward or downward. And if the tendon was not elastic, which is not the case, it would be torn. So the fact that the tendons are elastic is protective to them against tears. What do tendons consist of other than collagen and other than the fibrous connective tissue? They contain, like any living tissue, they contain blood vessels and they contain nerves, even if this is not written on the slide. The importance of the blood vessels is that they provide oxygen and nutrients for the molecules of collagen and the connective tissue fibers inside the tendon and carry out or carry away the waste substances. Of course, the importance of the nerve fibers that do not figure here on the slide is that they allow you to feel pain whenever there is excessive stretching on the tendon or excessive strain or on the tendon, resulting in a tendon sprain, for example, and in such a case, a strain, and in such a case, if you don't feel pain, this would be injurious to the tendon and the tendon will be torn. You might also ask about the abundance of blood vessels in the tendons. the blood vessels are not that abundant in the tendons, and this is the cause of slow repair whenever the tendon is injured. So are there blood vessels in the tendons? Yes. Are they abundant? No. And this justifies or explains the slow repair whenever you have a tear in the tendon. The importance of the blood vessels in the tendon is not only to carry oxygen and nutrients, which is number one for the sake of production of energy, nor to carry away waste substances only, which is number two, but also to help repair of the tendon whenever there is injury. So actually, the blood vessels have three functions, and the nerves also have an important function, which is not only feeling pain and feeling stretch, but protecting the tendon against excessive stretch. So tendons are supposed to be flexible and elastic. I might ask you why. Tendons are flexible and elastic because they are rich in collagen, which is an elastic protein molecule. I might also ask you about the importance of the tendons being flexible and elastic. When the tendons are flexible and elastic, this helps them to be stretched without being torn, and also helps them to bounce back after being stretched to the original position and their original length. But sometimes I might ask you what happens when? What happens when the tendon is exposed to severe stress? When the tendon is exposed to severe stress, however flexible and elastic it is, this will result in inflammation because the tissues are being exposed to high tension force that stretches out the components or the molecules of collagen inside and the connective tissue fibers in general, to the extent that they get damaged. And when certain cells in the connective tissue, like mast cells, for example, get damaged, they release histamine. And histamine, as you know from grade 10, is a chemical mediator that initiates inflammation. So when the tendon is excessively stretched, to the extent that damage occurs to mast cells inside the connective tissue, inflammation occurs. So you will find the area red, swollen, and warm and tender or painful. And these are the four cardinal signs of inflammation. Sometimes the stretch is severe enough to cause a tear in the tendon. So the first degree of or the first result of stretch is inflammation. But if the stretch is extensive, you will also find tear, and tear will intensify the inflammation. Pain is one of the symptoms of tearing of the tendon or of a torn tendon, because as we said before, whenever there is a tear, there is damage to the mast cells. And whenever there is damage to the mast cells, there will be inflammation. So inflammation can result from just stretching of the tendon or from an actual tear of the tendon. And in both cases, we will have the signs, the cardinal signs of inflammation, which are tenderness, redness, swelling, and heat or warmth. And as we said before, the process of healing in the tendons is very slow when they're left on their own, meaning that if we do not interfere, if we do not take any medical or surgical measures, then the tendon will heal on its own. And what happens when the tendon heals on its own? It would be a slow process of healing. Why is the process of healing slow in tendons? Because they have a relatively low blood supply. How can we treat torn tendons? Just like in any other case where surgical intervention is possible, we usually start by the non-surgical treatment or the medical treatment. In case of a torn tendon, the non-surgical treatment consists of 4 steps. First of all, RICE. And RICE is an acronym that stands for rest, ice, compression, and elevation. Of course, the main purpose of all these 4 components of RICE is to minimize the blood flow to the tendon for a while in order to minimize the swelling, the heat, and the pain. But of course, we need the blood flow to the tendon. But for a while, we have to rest to diminish exertion and to diminish blood flow to the muscles. We have to apply ice because ice causes vasoconstriction or narrowing of the arterioles and blood capillaries, and therefore will diminish the process of swelling, compression, which will result in the same effect, and elevation, because when you elevate the limb, you're elevating it against gravity, and therefore the blood flow to the limb will diminish. All these measures are temporary and they help diminution of the blood flow to the place that is inflamed for a while. And then we can fix with a brace for a while if the above measures are not successful, and it's for the same purpose, to minimize the stress on the tendon. And then we can use anti-inflammatory drugs like non-steroidal anti-inflammatory drugs or even steroids in severe cases in order to diminish inflammation. Because when you diminish inflammation, you diminish the swelling, you diminish, therefore, the pain. Physical therapy is indicated only when the acute symptoms have disappeared, meaning that you cannot prescribe physical therapy to a patient who is still under severe pain or who is still in the acute stage of inflammation. If all these measures do not succeed in improving the symptoms and improving the function, we have to interfere surgically, because if you don't interfere with a minor surgery, then the patient, every time that the patient moves the limb or the joint where the inflamed tendon is present, he will feel pain, and this will minimize the range of movement of the joint, will minimize the work of the muscles surrounding the joint, and might result in atrophy of the surrounding joints. Therefore, if the patient can withstand pain, and if the threshold of pain for the patient is high, this doesn't mean that you will leave the patient as he is or as they are without surgical intervention. So if you have a large tear in the tendon that is more than 3 centimeters and that persisted for more than 3 to 6 months, or if signs of muscle weakness start to appear, then you have to interfere with a minor surgery where you make a small cut in the skin over the tendon in order for the tendon to be accessible to you. And then you hold the torn ends of the tendon and you stitch them together. But sometimes, especially if the tear has been there for long, there might be some kind of contracture or shortening of the muscle, and the torn segments might be too far from each other to be stitched, and in such a case we might need a graft or a piece of tissue to put in between the torn ends so that to approximate them together and be able to stitch. We call this a tendon graft, and of course we do not take the tendon graft from another person, but from the same person, in order to avoid rejection of the graft and in order to avoid allergic reactions. Ligaments are very important. Ligaments are not part of the muscular system, but they are part of the musculoskeletal system. The ligaments differ from tendons because, as we said before, the tendon is nothing but the tapered end of the epimysium. So the tendon is part of the muscle that attaches it to the bone, but the ligament is not part of the muscle. So it's not part of the muscular system, but it's closely related to the muscular system because the ligament helps stabilize the joints that are being moved by the muscles. And that's why we're studying the ligaments. Ligaments are made of connective tissue, just like tendons are. And ligaments, the connective tissue in the ligament is full of collagen, just like the connective tissue in the tendon is full of collagen. Therefore, ligaments are elastic. But the main difference is that ligaments connect bone to bone, whereas the tendons connect muscles to bone. Ligaments are mostly flattened or flat, whereas the tendons are mostly rounded in their cross section. The importance of the ligaments is, number one, they hold the two or more bones that form the joint together, and therefore they help stability of those bones. They help stability of the joint that is made by the bones. And by holding the bones together and providing stability to the joint, they prevent displacement of the bones over each other and therefore minimize the friction. Because imagine that the two bones weren't stabilized by the ligament. In such a case, with every movement of the joint, they would move back and forth or upward and downward, and there would be friction in between them, or there would be a greatest wear and tear over the joint. It's true that there are other cartilages, like this articular cartilage and this meniscus, or those menisci, but as time passes, the wear and tear, the repeated use, would result in erosion of those cartilages. So the importance of the ligaments, say for example, this is the knee joint. This is the knee joint anterior view, and this is the knee joint posterior view. As you can see, a joint is the place where the end of one bone comes close to the end of another bone. In case of the knee joint, it is this bone, which is the lower end of the femur and the femoral condyles, or these two processes that form a joint with the upper end of the tibia. But this small lateral bone, which is the fibula, does not enter in the formation of the joint cavity of the knee joint, or it does not form the knee joint. As you can see, we have here the cruciate ligament. So we have the anterior cruciate ligament that connects the lower end of the femur with the upper end of the tibia anteriorly. And we have the posterior cruciate ligament, which is this one, and that we can see here. And that connects the same two bones, but from their posterior aspect. And because the anterior cruciate ligament and the posterior cruciate ligament are almost at right angles to each other, they form like a crucifix or a plus sign, and that's why we call it the cruciate ligament. Now, so the importance of the cruciate ligament is that it holds the lower end of the femur with the upper end of the tibia, and that it helps stabilization of the knee joint, and therefore it facilitates movements of the knee joint and minimizes friction. But more importantly, for minimizing friction, we have the cartilages. And the cartilage is a smooth elastic tissue that separates the articular surfaces of bones. And we have two types of cartilage inside the cavity of the knee joint. Why am I saying all the time the cavity of the knee joint? Because there is a structure that is not shown here that is called the capsule, which is a connective tissue capsule that surrounds the knee joint from outside and inside the capsule. That does not figure here, that we cannot see here. There is fluid called the synovial fluid. Of course, we do not study it here in detail because we are not studying the skeletal system and the joints in detail. So when I say the joint cavity, I actually mean the joint. Now we have two types of cartilage here. The first one is the articular cartilage that covers the lower end of the femur and the upper end of the tibia. But the articular cartilage here, which shares in the formation of the articular surfaces or the joint surfaces, is not seen here because it's hidden by the two menisci, but we can clearly see the articular cartilage at the lower end of the femur from anterior and from posterior. So first of all, we have the articular cartilage, and second, we have the two C-shaped cartilages that are called the menisci. The one towards the middle line is called the medial meniscus, and the one towards the outside, because the fibula is lateral, is called the lateral meniscus. For the articular cartilages, they just prevent friction between the bones. But for the menisci, they also prevent friction between the articular surfaces of the bone, but they also have two other important functions. The first one is that they absorb mechanical shocks whenever you jump, for example, or you go downstairs or upstairs. And more than this, they distribute the weight of your body over the whole articular surface to minimize injury. What happens if the cruciate ligament is torn? This will depend upon which part of the cruciate ligament will be torn. If it is the anterior cruciate ligament that is torn, then it is mainly anterior displacement of the femur over the tibia. And if it is the posterior cruciate ligament, then it will be posterior displacement. Now, as we know, the skeletal muscles, like the biceps muscles, are essentially voluntary, but they might be involved in some involuntary actions, like in case of a reflex action. What is a reflex action? And it's important here to highlight that there is a difference between the term reflex action and the term reflex arc. The reflex arc is the pathway that you see here, the arc that you see here, the pathway of the electric impulse. But the reflex action is the response itself. It is an automatic involuntary response that is carried out by a skeletal muscle or by a smooth muscle, and it is usually protective to the body. It might have its center in the brain, or it might have its center in the spinal cord. But when it has its center in the spinal cord, it is the spinal cord that makes the decision, but afterwards, the spinal cord sends an ascending sensory fiber to the sensory center in the brain to inform it about the sensation we have perceived. Now, if we look at the pathway here, we start the reflex action by a stimulus, which is a change in the external environment. And here, the change in the external environment is the heat that is given by the candle. And then we have receptors in the skin. And receptors, by definition, are what? They are modified sensory nerve endings. Is it enough to define a receptor by saying it is a modified sensory nerve ending? No, you have to say that it's a modified sensory nerve ending that is capable of changing the heat stimulus here, or the heat energy of the stimulus here, into electric impulse. And actually, every type of receptor is capable of converting or transducing a certain type of energy into electric energy and electric impulses. These electric impulses will be carried along a sensory afferent nerve. Afferent means towards the spinal cord, and then you have inside the spinal cord an intercommunicating neuron, or a relay neuron, that is sometimes not present in order to make the reflex more rapid. And then the relay neuron transmits or transfers or carries the impulses from the sensory neuron to the motor neuron. And then the motor neuron releases a neurotransmitter at the synapse or the neuromuscular junction, and then it stimulates the muscle to contract. A muscle cannot contract without having received an electric stimulus. From the relay neuron, there is an ascending sensory neuron that will later on, after a fraction of a second, carry the information, the sensory info, to the sensory center in the brain so that you can feel the heat. Of course, if you injure, if you damage the receptor, or if you damage the sensory neuron, the reflex will stop. The reflex will stop, and you wouldn't feel the sensation because the ascending fibers would not receive any stimulus. If you injure the motor neuron, then the muscle, of course, will not contract involuntarily, and the reflex will stop. Of course, this neuron that starts at the anterior horn cell of the spinal cord is the lower motor neuron. So any kind of injury to this motor neuron will result in not only in losing the reflex, but will also result in a lower motor neuron lesion in which the muscle will be flaccid, and there will be hyporeflexia, or there wouldn't be reflexes at all. Now, this diagram is very important because it shows you a section in the two cerebral hemispheres. It shows you the motor center in the brain and how we have descending motor fibers from the motor center in the brain that descend on the same side and then that cross to the opposite side, and then that cross to the opposite side at the medulla oblongata, or at a site in the medulla called the pyramid, and then they descend on the opposite side to reach the skeletal muscle on the contralateral side. This is the descending motor neuron, or the upper motor neuron. So what is the upper motor neuron? It is the motor neuron that starts in the motor center in the brain and that descends until it reaches the medulla oblongata and crosses to the opposite side to supply the muscle. Of course, a few of them descend on the same side, but for the time being, and for your curriculum in grade 11 of human biology, we will consider only those that cross to the opposite side. So if there is a lesion here in the brain center, what happens? First of all, the lesion can be due to an infarct, a clot, an occlusion of the blood supply, or it might be due to cerebral hemorrhage. For example, if the patient has a sudden elevation of the blood pressure, or because of an accidental trauma, for example. So if the motor neuron is injured here in the brain center, or if it is injured here above the decussation, then the muscle that will be affected and paralyzed will be on the contralateral side or on the opposite side. So if you have a brain stroke on the right side, on the right cerebral hemisphere, then there will be hemiplegia, because all the muscles on the opposite side of the body will be affected, the skeletal muscles. And in such a case, the patient will have paralysis of the contralateral half of the body, and that paralysis is an upper motor neuron paralysis, and it is characterized by two important characteristics. The first one is the hypertonia, the increased tone or spasticity of the muscles, and the second characteristic is the hyperreflexia, or the exaggerated reflex. Okay, these are the upper motor neurons, or the descending motor neurons. And we've talked about the lower motor neurons that start at the anterior horn cell when we were talking about the reflex on the previous slide. The smooth muscles, as we said, are present everywhere. They are present in the esophagus, they are present in the stomach, they are present in the wall of the small intestine, in the wall of the large intestine, and they are mainly here for peristalsis and sweeping down food. In addition to this, in the stomach, we have churning, of the food contents forcibly and making chyme. In the large intestine, we have segmentation that, in addition to peristalsis that we studied in grade 10, and that's important for moving food back and forth in the segments of the large intestine to mix them well with water and to facilitate the formation of the feces for defecation. In case of the urinary system, we have the smooth muscles of the ureter that undergo peristalsis to push down urine to the urinary bladder, and then we have the smooth muscles in the wall of the wall of the urinary bladder. They are partly smooth and partly striated, but we also have the urethral sphincter. The urethral sphincter is mainly formed of voluntary muscles, and it is under our voluntary control, so you can prevent urination whenever you want, whenever the conditions are unsuitable. But before potty training and when you were just an untrained infant below 1 year of age, the process of micturition was just a reflex when the walls of the urinary bladder stretched were stretched out with urine. It's a spinal reflex, by the way, having its center in the spinal cord, and there was reflex contraction of the walls of the urinary bladder, and reflex relaxation of the urethral sphincter to release urine. We also have smooth muscles in the wall of the uterus in females, and see how the uterus is above the urinary bladder, and the contractions of the uterine muscles during labor or childbirth, or the contraction of the smooth uterine muscles during menstruation to expel the unwanted mucosa and blood in the uterus, are the cause of the severe pain the female senses or feels during the menstrual cycle, during menstruation actually, and during labor. The characteristics of the smooth muscles, as we said, they are unstriated muscles. They form broad sheets of muscles that are surrounded by connective tissue. But one important difference here is that the smooth muscles are not arranged in the form of muscle bundles and tendons and muscles having tendons. So the connective tissue sheets around the smooth muscle sheets do not taper to form tendons. As for the individual muscle cell, smooth muscle cell, it is tapered from both sides, and that's why we describe it under the light microscope and under the electron microscope as being spindle-shaped. It has a single central nucleus. The smooth muscle fibers are interlacing, and they form large sheets of muscles. And these muscles might be arranged in a longitudinal way, like the outermost wall of the stomach, or in a circular ring-like shape, like in the middle wall of the stomach, or in the sphincters, the cardiac and pyloric sphincters, as you studied in grade 10, or they might be arranged obliquely. So this terminology of having longitudinal or circular or oblique muscles is just related to the direction of the long axis of the smooth muscle sheet in relation to the axis of the organ. Of course, all smooth muscles are, or the majority of the smooth muscles are involuntary muscles. For the cardiac muscles, functionally, they are involuntary, but as we said before, they're striated because they have regularly arranged actin and myosin. Each cardiac muscle has one central nucleus, just like the smooth muscles do. The cardiac muscles, as we said before, have a peculiar anatomical or histological structure, which is the branches, and the fact that they are interconnected, and they have in between their branches, they have a structural connection and a functional connection. The structure, and both lie in the intercalated discs. So the intercalated discs are what? The intercalated disc is the thickened structure that lies at the junction of two branches. For example, here we have an intercalated disc. The intercalated disc is a thickened structure at the junction of two branches. And inside the intercalated disc, we have structural connection and functional connection. The structural connection is nothing but the filaments or the protein molecules called the desmosomes that look like threads and that prevent stretching, that prevent the cardiac muscles from being separated from each other when they are stretched out. But we also have the functional connection, and the functional connection is in the form of the gap junctions that contain the ion channels that allow the rapid transfer of ions and therefore the rapid transfer of electric impulses so that the cardiac muscles can contract as one unit and act as if they were a functional syncytium. Now, inside the cardiac muscles, we have specialized cardiac muscle fibers. Examples of these specialized cardiac muscle fibers are the sinoatrial node, the atrioventricular node, the bundle of His, the right bundle branch, and the left bundle branch. So these four structures that we have studied are specialized cardiac muscle fibers. Actually, there are five: SA node, AV node, His bundle, right bundle branch, and left bundle branch. They are not nervous tissue. They are modified cardiac muscle tissues. And these modified cardiac muscle tissues are capable of generating electric impulses spontaneously. But why then aren't they all pacemakers of the heart? Because the SA node, in addition to being able to spontaneously generate electric impulses, it does this at the highest intrinsic rate. So if I ever ask you, what does it mean that the SA node is the pacemaker of the heart? You would just say that the SA node controls the rate of the whole heart. But if I ask you about the structure function adaptation of the SA node, or what are the characteristics that make the SA node be the pacemaker of the heart, you would say that it is a modified cardiac tissue that can generate electric impulses spontaneously at the highest intrinsic rate, which is 60 to 90 beats per minute. And I might ask you further, why is it capable of generating electric impulses spontaneously? And you would tell me that it is because it has an unstable membrane potential. The AV node is another modified cardiac muscle tissue that lies at the junction of the atria and ventricles. That's why it is called atrioventricular node. Whereas the SA node is located where? The SA node is located in the right atrium at the posterior wall, embedded in the posterior wall of the right atrium. The AV node is the deputy of the SA node, meaning that the AV node can generate electric impulses spontaneously and therefore can act as the pacemaker of the heart, but only in the absence of the SA node, only when the SA node is sick. Why? Because it's true that the AV node can generate its own electric impulses or can generate impulses on its own, but at a slower rate, which is 40 to 60 beats per minute, which is usually not enough to support the pumping action of the heart. So what are the functions of the AV node? Number one, it acts as a spare pacemaker if the SA node is sick or not functioning. Number two, it relays and delays. It relays, meaning that it transmits impulses from the atria to the ventricles. Number three, it delays because of its slow rate of firing of electric impulses. It causes slow conduction between the atria and the ventricles. And this is important for two things. Number one, to prevent contraction of the atria and the ventricles at the same time, which is simultaneous contraction. And number two, to allow the ventricles enough time to fill with blood before pumping. As for Bachmann's bundle and for the moderator band, we didn't study them. But if you want to know the function of the Bachmann's bundle, which is also a modified cardiac muscle tissue, it allows the rapid transfer of electric impulses over the bulk of the atrial muscles, whereas the moderator band prevents excessive dilatation of the right ventricle. The His bundle is important because after the AV node makes sure that the ventricles that the ventricles will not contract at the same time as the atria, the bundle of His will rapidly conduct the impulses to the ventricles, and therefore its two branches, the right branch of the bundle of His and the left branch of the bundle of His, will also transfer the impulses rapidly to the right and left ventricle almost at the same time, almost simultaneously, in order to allow efficient contraction and efficient pumping of the ventricles. The right ventricle will pump to the lungs deoxygenated blood to be purified, to get rid of the carbon dioxide and be loaded with oxygen, whereas the left ventricle will pump oxygenated blood to the whole body. Now, in this diagram, you can see that every myofiber is supplied by a nerve fiber and has its own synapse and its own myoneural junction. So the more you stimulate, the higher the intensity of the electric impulse in the nerve that supplies the whole muscle, the more the number of individual muscle fibers that will be stimulated, and therefore the greater the power of contraction of the muscle. So the strength of muscle contraction depends upon the number of nerve fibers that have been stimulated, and therefore upon the number of myofibers or muscle cells that will contract. But before saying this, I would rather say that when you increase the intensity of the electric impulse above the threshold stimulus, but below the overload stimulus, you will increase the strength of muscle contraction because you're actually increasing the number of myofibers that are contracting. But as for individual muscle fibers, every muscle fiber obeys the all or none rule. Of course, here there is some important information that every muscle should receive enough blood and should receive a nerve supply, and that blood is important to carry oxygen, nutrients, to give energy in the form of ATP molecules that are needed for myosin heads to attach and to detach. The muscle also needs veins because the veins are the ones, the blood vessels that carry out waste. Is it important to carry away waste? Yes, of course, because if the waste accumulate in the muscle, it would interfere with muscle function. Coming back to the all or none rule, as we said before, every muscle fiber either contracts maximally or does not contract at all. And in order for the muscle fiber to contract maximally, it should receive a threshold stimulus at least. What is the threshold stimulus? The threshold stimulus is the minimum stimulus or electric intensity that is capable of stimulating a muscle and causing its contraction. Here you can see the actin and myosin filaments. These are the smallest subcellular structures, and they form part of the myofibrils. So the myofibrils, the spaghetti-like structures, the striated spaghetti-like structures in the sarcoplasm of the muscle, actually consist of smaller molecules that are called the myofilaments, as we said before. And a filament is a a filament is a protein molecule. So we have the thin actin filaments that are formed of beads, and the thick myosin filaments that are formed of parallel protein filaments, but that also have these spherical heads that arise from them. And here you can find that we have this pink molecule that covers the binding sites, and calcium is important here, calcium ions, to remove that pink molecule that we haven't studied or whose name hasn't been mentioned in class, and allow the myosin heads to bind with their actin binding sites. As you can see here, we have ADP, which results from hydrolysis of ATP. So in order for the myosin head to attach with actin, to attach to actin sites, it has to hydrolyze a molecule of ATP into ADP plus phosphate plus energy. And this energy will be used by the myosin head to strike the actin filaments and to pull on them to drive them towards the center of the sarcomere. And this is according to the sliding filament theory. So in order for myosin to attach, it needs ATP. So if you don't have enough ATP, there wouldn't be contraction of the muscle, or contraction of the muscle would be weaker than normal, or the muscle wouldn't be able to contract as normal. Not enough muscle fibers will contract, so the strength of muscle contraction will diminish, and this is muscle fatigue. So what is muscle fatigue? It is a state where the muscle cannot carry out its normal functions as usual because of the depletion of ATP, but because of the relative depletion of ATP. But sometimes when there is severe depletion of ATP, it's not only that the myosin heads cannot attach to actin, but they do not find enough ATP to detach from actin, and the muscle stays in a state of continuous weak contraction, which is tetany. So tetany is a status where the muscle is in a continuous state of contraction, either because you don't have enough ATP molecules, so the myosin heads do not detach and keep attached to the actin or stay attached to actin, or another cause of tetany can be hypocalcemia, diminution of calcium ions in the blood, because calcium is important for stability of the muscle and diminished excitation of the muscle, or because of the third cause of tetany, it might be a neurotoxin, like in case of Clostridium tetani, or number 4, it might be done in the lab, or you might cause it to yourself if there is repeated frequent stimulation of the muscle. So we've just mentioned 4 causes of tetanic contractions, or continuous contraction of the muscle, which is usually very painful. It might be due to depletion of ATP and failure of myosin to detach from actin. It might be due to a neurotoxin, like in case of Clostridium tetani. It might be due to hypocalcemia or diminished calcium ions in the blood. And finally, it might be due to repeated frequent stimulation of the muscle, and you can do this in the lab using special instruments. In case of hypocalcemia, the treatment of tetany is to give an infusion, an intravenous infusion of calcium, but it has to be very, very slow, because if you give calcium rapidly, this might result in death of the patient because of a sudden drop in the blood pressure, because of a shock that occurs to the patient, and rarely because of tetanic contraction of the heart. Because just like hypocalcemia causes tetany, hypercalcemia might cause tetany. But luckily, in case of the heart, tetany is very rare, because the cardiac muscles, as we said in the classroom, have a very long absolute refractory period. What is the absolute refractory period? Refractory means irresponsive. and absolute means totally irresponsive, whatever the strength or the intensity of the stimulus is. So if tetany can occur in skeletal muscles, it's absolutely rare in case of the cardiac muscles. Now, if we want to explain muscular contraction on molecular basis, this means that we have to examine the muscle under the electron microscope and explain the sliding filament theory. So if I ever ask you to explain the contraction and shortening of the muscles on molecular basis, then I'm actually asking you to explain the sliding filament theory. So you're going to talk about the sarcomeres, and the sarcomeres are the functional unit of a skeletal muscle or a cardiac muscle, meaning that every sarcomere alone can carry out the function of the whole muscle. In every sarcomere, we find that it's bonded by two Z discs that are made of thin filaments. The actin filaments are parallel to each other. They arise on each side at the Z line and extend towards the center of the sarcomere. But there is a part in the center that does not contain actin. Myosin filaments, the thick filaments, alternate with actin. So here we have a very regular arrangement that is responsible for the striations. We have a dark band, which is the A band, and a light band, which is the I band. The A band, or the dark band, is the place where you can find both actin and myosin, So it extends from here to there. And in the middle of the A band, or the dark band, you have this H zone that is dark, but less dark than the rest because it contains myosin only, but not actin. On the other hand, the I band contains only actin filaments. One half of the I band is present in one sarcomere, and the other half is present in the other sarcomere. I wouldn't ask you about the A band and the I band on the test, but I can ask you about the striations, and you would simply say that the striations are due to the presence of a regular arrangement of alternating thick myosin and thin filaments of actin. So again, I want to see these three descriptions: a regular arrangement of alternating. So regular is number one, alternating number two, thick myosin filaments number three, and thin actin filaments number four. And then the sliding filament theory states that upon giving ATP to the muscle, upon hydrolyzing ATP, the myosin heads attach to specific binding sites on actin and pulling them towards the middle. So actually the actin filaments slide on top of the myosin filaments. And when they do so, they approximate the Z discs together, or the Z lines together, meaning that the Z lines come close to each other and the sarcomere shortens. And since every myofibril is made of a series of sarcomeres, so if all the sarcomeres shorten, then the whole muscle shortens. And this is the cause why a muscle like the biceps thickens upon contraction, because it shortens. And this is also the cause why it can move a joint, because it approximates the insertion, which is attached to the movable bone, towards the origin, which is attached towards the stationary bone. So this sliding filament theory explains what? It explains shortening of the muscles upon contraction, thickening of some muscles upon contraction, and getting the insertion closer to the origin. We have mentioned all this, and of course we have talked about the antagonistic muscles. We also talked about the importance of ATP. Just like all cellular activities, ATP is necessary for muscle contraction, because As we said before, ATP is important for attachment of myosin, and ATP is important for detachment of myosin. In each case, whether myosin is attaching to actin or detaching from actin, there is a molecule of ATP that is hydrolyzed by the addition of water, that's broken down by the addition of water into ADP and inorganic phosphate, and energy is released. So the process of hydrolysis of ATP is an exothermic process because it results in the release of the stored chemical energy in the ATP molecule, and this energy will be utilized by the myosin heads to attach or to detach. So what happens if the muscle is depleted from ATP? There will be muscle fatigue and maybe also muscle tetany. Muscle fatigue because myosin attachment to actin will not occur properly, and therefore sliding and shortening of the muscle will not occur properly. Therefore, this results in the muscle not carrying out its function properly, and it might also result in a continuous contraction because the deficiency of ATP would result in deficient detachment of the myosin heads. But fatigue is not only due to depletion of ATP and failure of actin to slide over myosin, but it's also because of something else, which is the production of lactic acid. Because as you know, when the muscle does not receive enough oxygen, or when the muscle has a relative deficiency of oxygen, it will undergo anaerobic cellular respiration and produce lactic acid in a process called acidic fermentation. And lactic acid would stimulate the nerve endings and cause pain, cramps, and soreness. And we will be talking about this in detail very soon. Of course, this is the chemical structure of ATP. Here we have adenine, which is a nitrogenous base, ribose sugar, and both are present, by the way, in RNA. And then we have 3 phosphate groups. So this is adenosine triphosphate. And as you know, the bond between the last phosphate group and the remainder of the molecule is a high energy bond. When you add water, you break down that bond. So 1 phosphate is released, and the remainder is adenosine, which is adenine and ribose, diphosphate. But by having broken this high energy bond and having released this inorganic phosphate, there is a good deal of energy that is released and that is utilized in contraction of the muscle. We have mentioned the all or none rule, and that it is applicable to a single myofiber, but the whole muscle intensity of contraction can increase or decrease according to how many myofibers are stimulated. We have also talked about the muscle tone. The muscle tone is very close to tetany because the muscle tone is a continuous state of contraction. But the differences are that, number one, what are the differences? Number one, that the tone is something physiological. It's a normal process that is useful to the body, whereas tetany is a pathological process that is harmful to the body, associated with diseases, and might result in loss of function of the muscle. The muscle tone is a partial contraction, a mild contraction, a weak contraction. And this contraction is here just to either give shape to the body or to stabilize the body in its erect or upright position, just like the anti-gravity muscles do. The anti-gravity muscles are the muscles of the back, and all types of muscles that antagonize gravity, that prevent falling forward of the human body or leaning forward of the human body. The anti-gravity muscles, as long as you're standing up in an upright position, the anti-gravity muscles are working and they have a tone that's higher than the tone of other muscles, that muscles of the front, for example, that do not antagonize gravity. But why don't they get fatigued? Because they alternate. They do not receive electric impulses at the same time. For example, if I have 100 muscle fibers at the back, so 30 or 50 will be contracting and 50 will be relaxing, and then they will alternate with each other. Muscle cramps, as we said, occur when there is severe depletion of energy, when the muscle is depleted from ATP and therefore the myosin heads cannot detach, and when there is relative deficiency of oxygen to the muscle, resulting in the muscle referring to anaerobic respiration, which is, in the case of the muscle, acidic fermentation, producing lactic acid that causes the cramps, pain, and soreness. I've repeated several times relative oxygen deficiency. What do we mean by relative oxygen deficiency? This occurs in case of strenuous muscle exercise when you have exercised for a long period of time. So the heart is beating maximally to pump blood maximally to the muscles, and the lungs are breathing at the maximum possible physiological rate to provide this blood with oxygen and to carry it to the muscles. However, despite this maximum supply of oxygenated blood to the muscle, the needs of the muscle are more. This is the condition of relative oxygen deficiency that results in the oxygen debt. So the oxygen debt is when you have relative oxygen deficiency. You are receiving oxygen maximally. You are producing ATP for the muscles at a maximum rate. The heart is doing its maximum. The lungs are doing their maximum. However, you don't have enough ATP for the muscle to contract, and therefore you are in a state of oxygen debt. What do you do at that time? You refer to anaerobic respiration because it does not contain oxygen. So you do acidic fermentation. You get some molecules of energy that are less, because in aerobic you get 38 ATPs per glucose molecule, but in anaerobic you get only 2 ATPs per glucose molecule. However, you get some ATP to finish your exercise, but you are in a state of debt because you are producing lactic acid. So after you finish your exercise, you keep breathing rapidly, and you always have to justify this. Why do you keep breathing rapidly? Because all this lactic acid that has accumulated has to disappear. And in order for it to disappear, there are two things that have to be done by the human body. The first thing is to wash it away by the venous return, and that occurs normally, but there is something else that has to be done, is to reoxidize all that lactic acid in order to avoid its harmful effects on the nerves and the pain and cramps and the soreness it causes. So you keep breathing rapidly in order to get it more oxygen, despite you finished your exercise and despite you're resting now, just to obtain more oxygen to oxidize the lactic acid and get rid of the harmful symptoms. Here we have the sources of energy for the muscle contraction. There is a certain hierarchy. We start by the glucose that is available in the blood, and if all the glucose in the blood is terminated, we refer to breaking down the glycogen in the muscles. Whether you are obtaining glucose from the blood directly or from the breakdown of glycogen, you go through aerobic respiration. And aerobic respiration passes through two processes, glycolysis and oxidative phosphorylation. You do not need to memorize these two terms in the blue boxes, but you have to know that aerobic cellular respiration produces a good deal of ATP, and that this ATP has to be hydrolyzed, as we said, And we have two enzymes to hydrolyze ATP. An enzyme to hydrolyze ATP for contraction, for attachment of the myosin heads, and another enzyme to break down ATP for relaxation or detachment of the myosin heads. First one is myosin ATPase, and the second one is calcium ATPase. And an ATPase is an enzyme that hydrolyzes ATP or breaks it down in the presence of water. You do not need to know the names of enzymes, nor to know glycolysis and oxidative phosphorylation, but you have to know that the first source of fuel for the muscle is glucose. The second one is glycogen, when all glucose is depleted and that glycogen is converted or hydrolyzed into glucose, that they both go through aerobic respiration to produce ATP, that ATP has to be hydrolyzed once for contraction or attachment of ATP heads, of myosin heads to actin, and once for relaxation or detachment of the myosin heads. What's the importance of creatine phosphate to the muscle? Or what's the importance of creatine to the muscle? When creatine reaches the muscle cells, it attaches to the phosphate from the phosphate pool in the muscle cell, and it forms creatine phosphate. The importance of creatine phosphate is that it releases phosphate very easily and very rapidly, and therefore creatine phosphate releases phosphate easily and is important for resynthesis of ATP and replenishing the stores of ATP. Of course, oxygen is important here for aerobic respiration. If oxygen is not present, then after glycolysis, the muscle will carry out acidic fermentation and produce this harmful lactic acid. If we have depleted all our glucose and glycogen stores and all our ATP stores, and we want to replenish our ATP, then the last resort would be the fats under the skin or the fatty acids in the blood. And these fatty acids will go to the mitochondrion, join aerobic respiration, and produce ATP. But this is the last resort for the cell, because this oxidation of fatty acids can produce harmful byproducts for the cell. We have mentioned muscle fatigue, which is the physiological inability of the muscle to contract, or the inability of the muscle to contract in a normal way because of a relative depletion of ATP. And we now know what relative means. Relative means that we have a lot of ATP, but it's yet not sufficient for muscle contraction and relaxation because we are working out maximally, okay? And these are the energy resources, and we talked before about the antagonistic muscle groups. I have something important here to say because I've seen some mistakes. When I say that the biceps and the triceps are antagonistic, this means that when each of them contracts, it does the opposite of the other. So when the biceps contracts, it flexes the elbow joint. When the triceps contracts, it extends the elbow joint. Flexes means bends. But when the biceps contracts to flex the elbow joint, the triceps must relax. The triceps has to relax, because if the triceps does not relax and keeps contracting, the biceps wouldn't be able to flex the elbow joint. On the other hand, if we want to extend or straighten the elbow joint, it is the triceps that has to contract. And in that case, the biceps must relax, because if the biceps does not relax, then the triceps will not be able to straighten the elbow joint. It's also important to know the origin and insertion of the biceps and triceps. The biceps has two origins that are both on the scapula, and it has an insertion that is mainly inserted into the radius and partly into the ulna. The triceps has three heads that are attached also to the scapula, and the insertion is in the forearm bone, especially in the ulna, but from the posterior aspect. Muscle injuries, of course, are the last thing to say. A muscle bruise is resulting from a blunt injury to the muscle that results in inflammation, meaning that the bruised muscle will be swollen, will be hot or warm, will be painful, but there might be internal bleeding because it might be a blunt trauma that did not cause laceration or tear in the muscle, in the skin and in the muscles, but it caused rupture of some of the capillaries inside. So having intact muscle fibers does not mean that there is no internal bleeding because of rupture of some small capillaries. But a muscle laceration, on the other hand, is a tear in the muscle fibers because of a sharp trauma. Strain is a term that describes a muscle or a tendon when it is stretched more than it can afford, whereas a sprain results from overstretching or torsion or twisting of a ligament or a capsule, and a capsule is the connective tissue outer coat of the joint. Thank you so much, and I wish you good luck and the best of success on your test on Thursday, inshallah.